Okay, well, welcome back. We are here tonight to do the urinary system, and so I thought you would think that my introduction here was quite appropriate. So anyway, we will be talking about basically how the kidneys work and the production of urine. So I know you're all quite excited about that. Um, so when we talk about the urinary system, um, it's going to consist of the two kidneys that are really going to act as these filters for the blood. And then the two ureters, which I'll show you here in just a minute. Let me get my pen color so I can draw on the slides here. So again, and here's the kidneys. This is actually, you can see here. Oh, my pad there. You can see the kidneys, the urinary system was pulled out here. This is a cadaver urinary system. And so here's your two kidneys. Here's the two kidneys here. And then you have the two ureters. And the ureters are going to be these long tubes here that lead all the way down to the bladder. So they connect, basically, the ureters connect the kidneys to the bladder. And then here's, of course, the urinary bladder that's going to temporarily store the urine before it's expelled. And then the urethra, which you really can't see on either one of these, will be down here at the base of the bladder, and that's to convey the urine as a waste substance out to the outside of the body. So that gives you a quick overview of the major organs of the urinary system. So again, the kidney ends up being this kind of reddish-brown, bean-shaped organ. It's reddish-brown, as you're going to see, because it's got a lot of vascular flow through it, so it's got a lot of blood vessels in it. And it's typically about 12 centimeters long, just to give you an idea of how big it is. It's enclosed in this tough, fibrous capsule on the outside that we call the renal capsule. Okay, so the renal capsule encloses the kidneys. When we talk about the kidneys are actually located what we call retroperitoneally. And what that means is retro means behind, and then the peritoneal is the peritoneum is that serous membrane that covers the abdominal organ itself of a parietal, that the peritoneum that the organs is actually going to be in front of the kidneys. So the kidneys are behind the peritoneal cavity. They're outside that membrane. So sorry about that. I'm not sure what my Adobe is doing there. So again, the kidneys are what we call retroperitoneal. They're behind the peritoneal cavity, or behind the peritoneal membrane that's covering all those abdominal organs. And if you look again, the left kidney will actually be slightly higher than the right kidney. Okay, so let's get into some of the actual kidney structure. So again, if you look on the outside of the kidney here, on the outside you will see this is the renal capsule. Okay, so the renal capsule is going to be that fibrous capsule that covers the kidneys. Now when we look inside the kidneys, we're going to start to look at some different structures now. In a little bit we're going to talk about the fact that we have these basic functional units of the kidneys called a nephron. Okay? And these basic functional units of the kidneys, again called the nephrons, are going to primarily be mostly in the what we call renal medulla, but a portion of them will stick up into the renal cortex. So this is the renal cortex, guys. All of this layer right here is all the renal cortex. Okay? So when we're referring to renal cortex, especially later on when we get into talking about the nephron, I want you to go back to this picture if you need to remember what the renal cortex is. And then this here is going to be the renal medulla. Okay, and the renal medulla is going to be where the majority of your nephron is going to run through. So you're going to see the renal medulla actually consists mainly of the tubules of your nephrons. So again, this is just a little bit of our kidney anatomy. So renal cortex, renal medulla. Now, as things are happening in the nephron, and we're going to be talking about exactly what happens in the nephron. But as urine is formed in all these tubules and these nephrons, okay, eventually all of those tubules will come together in the medulla at what's called the basically the minor calyx. So you'll hear this called right down here the renal pyramid. And so all of these the collecting ducts of the nephron are going to come together here, okay, at the base of the medulla here, at what we call this renal pyramid, and dump into the minor calyx. Okay, so the nephrons, and you're going to see the very last portion of the nephron is something called the collecting duct. So this will make sense a little bit later when we cover the nephron. But the collecting duct of the collecting ducts of the nephron are all going to dump into, and again, there's, if you could picture thousands of them here, all dumping into the minor calyx. Okay, so we're going to be dumping that urine that's been produced into the minor calyx. Then the minor calyx will dump into the major calyx. Okay, so here these are all minor calyces, or what we call a calyx. And then here is the major calyx. And so again, then from the major calyx, we're going to dump into the renal pelvis. And so that's just saying that all of these, major calyxes are going to be dumping into the renal pelvis. That's going to be this big collecting area here. And then the renal pelvis will then directly flow into the ureters. Okay, that will then carry the urine down to the urinary bladder. So that's kind of the way that that's the pathway that urine is going to be produced and then moved out of the kidney. So again, it'll be produced in the nephrons, and then those will dump into the minor calyx, and then into the major calyx, into the renal pelvis, the ureters, and then into the urinary bladder. So make sure that you know the order, okay, in which urine is going to move into the urinary bladder and then out of the body. So again, that gives you kind of an overview. So this tells you there's a medial depression in the kidney that leads to what's called a hollow renal sinus, into which blood vessels, nerves, lymphatic vessels, and ureters enter. So this area right here is the renal sinus, and it includes the renal pelvis. So it's just saying the renal sinus is this area where the renal artery and the renal vein and any nerves are entering and leaving the area. So this whole area would be the renal sinus. Now the kidneys are going to function, again, their main function is going to be to regulate the volume, the composition, and the pH of body fluids. And oftentimes in terms of composition here, we mean kind of the makeup of like what kind of ions are going to be in the body fluid. So that's what we mean by composition. What kind of ions and solutes are you going to have in your body fluids? And then, of course, one of its other major functions is going to be to remove metabolic wastes from the blood in the process. So again, the kidneys are going to function to regulate volume. So they're going to regulate volume, composition, and pH of your body fluids. And then remove any of the metabolic waste from the blood in that process, while, again, they're regulating the compositions of your body fluids. The kidneys also are going to help control the rate of red blood cell formation by secreting that hormone erythropoietin. So remember erythropoietin from the last, the blood lecture. I know that might have been a little while ago for some of you. We also called it EPO. So we said that, EPO will be released especially when there's a lack of oxygen, so in the tissues. And so what happens is that erythropoietin, and sorry, it's unfortunately it should be TIN here. I didn't realize that it left off there. There we go, that many letters. There we go, there's erythropoietin. And so again, this is oftentimes abbreviated EPO. So what happens here is the kidneys, as blood flows through the kidneys, okay, the tissues inside the kidneys will constantly detect how much oxygen is in the blood. And if for some reason there is a lack of oxygen in the blood or low oxygen in the blood, then what happens is the kidneys will then secrete erythropoietin. So for example, if you went up to Flagstaff, okay, obviously the air is thinner up in higher altitudes, and so there's less oxygen available. And so what would happen was that your kidneys literally would detect the lack of oxygen, and they would start to produce erythropoietin. And erythropoietin, again, as a reminder, is that hormone that's going to increase red blood cell formation. So it'll stimulate red blood cell formation. So you literally make more red blood cells, which is the idea behind high altitude training, right? So that way then you make more red blood cells and then you come back down to a lower altitude and you still are going to have those extra red blood cells in your system for a few weeks. So you have a little bit of an advantage in terms of bringing oxygen to your tissues. So that's erythropoietin. And again, the kidneys are going to help obviously control the rate of red blood cell formation by just detecting again the oxygen and secreting erythropoietin. And then they can also regulate blood pressure by secreting a substance called renin. And so we're not going to get into too many of the nitty-gritties because this gets to be a complicated system, but I just want you to know that the kidneys also help to regulate your blood pressure, and they can do that by secreting this hormone renin. Renal blood vessels. So we're going to get into a little bit of the renal blood vessels, and I'm going to kind of break this down and tell you what you really need to know. Some of this is a little bit too much in terms of this is more of some of the information on the slide is more really Bio 201 that you would have to know this, or Bio 202. So this is an important slide because I'm going to tell you what you need to know and what you don't need to know. So the abdominal aorta. So remember the aorta coming off of the heart is going to branch, and eventually a portion of that aorta ends up becoming the abdominal aorta. And that abdominal aorta is going to give rise to renal arteries. So renal arteries are obviously going to be the arteries that are going to be entering the kidneys. And as the renal arteries pass into the kidneys, they're going to branch into these successively smaller and smaller and smaller arteries. Okay? Now, you do not have to know all these arteries. This is just to show you how much branching occurs. So again, you got segmental arteries branching into interlobar arteries, into arcular arteries, into interlobular arteries, and so on. You do not need to know all this. Okay? You are going to be responsible for knowing exactly how the blood flows through basically the glomerular apparatus, which is going to be part of the nephron. So you will be responsible for knowing this, and this will make more sense when we get into the nephron, so don't freak out too much now. But the afferent arterioles, the glomerulus, the efferent arterioles, and then the peritubular capillary. So let me just kind of give you an idea of what this looks like. And then, again, don't freak out too much about these last few terms because we're going to get into them. We're going to go over them. So this is just to show you, here's the renal artery. And again, the renal arteries are going to branch heavily, and you do not need to know all this branching. You don't need to know the segmental artery is branching into the lobar artery and into the interlobar arteries and then into the arcuate arteries and so on. You won't need to know all of that. But so here you can see, again, there's lots of branching, and then eventually they branch into these little capillaries. And then the veins here, eventually those capillaries will dump back into veins, and then you can see the veins here are going to basically go backwards and drain the blood back out of the kidney, okay, after again we've dumped oxygen and then picked up again a couple of, and you're going to see we're going to pick up certain ions and things in the kidneys as well into the blood, so and pick back up any ions and things that they need. So again, that blood will then leave the kidneys through the renal vein. So this is kind of nice because this slide, guys, is going to highlight what you need to know. Okay, so this shows you all of the blood vessels. I've basically bolded around all of the blood vessels that you need to know. So again, and the reason that you're going to need to know these is this is going to tell us how blood is going to flow into the nephron. So remember that it's at the nephron, okay, where the blood is actually going to be filtered. And this is showing you, here would be the nephron right here, and this is blowing the nephron up for you so you can see the nephron, and you can see that the blood capillaries are going to surround that nephron, okay? So what happens is that renal artery, again, is going to branch again into all of these that you don't have to know until eventually it will branch into what's called the afferent arteriole. This right here is the afferent arteriole right here, okay? And that afferent arteriole is going to be bringing blood into the nephron, okay, at the glomerulus. So here the afferent arteriole then is going to branch into this heavily branched glomerular capillaries. So we call this the glomerulus, this capillary bed right here that's inside this ball of the nephron is going to be called the glomerulus. So that's what we actually call those capillaries, that ball of capillaries, and it looks like this. Here's your afferent arteriole going in, and then all of a sudden the afferent arteriole is going to branch here, and it's going to branch like this. So to branch up here, 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 and it's this ball of capillaries, okay? And then eventually, okay, the blood will then come back out the efferent arteriole. So here, again, is your afferent arteriole bringing blood in, okay? And then, again, that afferent arteriole branches into all these capillaries, okay? And then eventually, the blood is brought back out through the efferent arteriole. So to help you remember, again, afferent arteriole and efferent arteriole, just think efferent, okay? Think of the word E there and remember the word exit, okay? So E, exit. So that is going to be taking blood and exiting the nephron, okay? So the afferent is bringing blood into the nephron. The efferent is taking blood away from or exiting the nephron, okay? If that helps you. So again, that's how the branching is going to occur. It goes from the afferent arteriole into the glomerular capillaries, okay, and then back out the efferent arterioles. Now, when it comes back out the efferent arteriole, then that efferent arteriole is going to run into what we call the peritubular capillaries, or also called the vasa recta. Same thing. Peritubular capillaries, vasa recta, guys, exact same thing. Just it depends on what book you have, on what term they choose. So here again, the peritubular capillaries, the vasa recta, are going to be these blood vessels that completely surround the rest of the nephron here. So the what we call the loop of Henle that we haven't talked about this yet, but we will in a minute when we get into the nephron. The peritubular capillaries are going to surround that loop there. And then eventually, those peritubular capillaries will then drain into your veins. So then they'll drain into the venous system. And then they'll take blood back out the kidneys through the renal vein. Okay? So that's how blood moves through the kidneys, in the renal artery, and then eventually into each of these nephrons here. Excuse me, my computer about to die, so I'm going to plug it in here. Sorry about that. But yeah, so you've got the renal artery branching into, again, these arteries, smaller arteries, into eventually arterioles, and then into glomerular capillaries here, and then into the efferent arteriole, then into these other capillaries, peritubular capillaries, and then finally, again, draining into the veins, okay, and out the renal vein. So that gives you a little idea of what's happening at the level of bringing, especially blood into and through the kidneys. And of course that's going to be very important because remember the main function of the kidneys is of course to filter blood. So one of their main functions, right? And so that's why the blood flow through the kidney is so important, and that's why when you look at the kidneys they look dark. The shape or the color of the kidneys is a dark color, and the reason it's a dark color is because of all those blood vessels running through the kidneys. So then again, venous blood is returned through a series of vessels and then eventually, okay, out the renal vein. So hopefully that pretty much covers how blood moves through the kidneys. And just keep in mind, I want you to keep in mind that as we get into talking about the nephrons, okay, keep in mind that when you actually look at the nephron, you know, we'll break it down and look at the nephron individually without the capillaries all over this. But remember that there are capillaries covering this nephron, and that's because we're going to be constantly absorbing things from the blood into the nephron, and then things are going to be moving from the nephron back into the blood as well. So when we're talking about that exchange that's going to be occurring, remember that can occur because there are these blood vessels that are just completely surrounding the nephron, these capillaries that are completely surrounding the nephron. So try to keep that visual image of that in mind when we get into the nephron. So now we're going to finally talk about the nephron. This will hopefully help fill in some of the gaps for you. So when we look at the nephron is the basic structural unit for the kidney. So basically, it is the unit that's doing all the work. Okay, the nephron is the unit of the kidney that is doing all of the work, all of the filtering action. And if you look at the kidney itself, the kidney contains over 1 million nephrons. So it contains, again, many of these nephrons, each of which of these nephrons consists of a renal corpuscle and a renal tubule. And we'll talk about the renal corpuscle coming up here, especially when we get into the glomerular apparatus here. And then the renal tubules, and then you can see all of these. These are all the renal tubules. And we'll break those renal tubules down and talk about exactly what each of these separate individual tubules are. So the renal corpuscle is going to be the main filtering portion of the nephron, okay? And so the ball up here is going to be the renal corpuscle, okay? It consists of a ball of capillaries called the glomerulus, okay? And a glomerular capsule, okay, that receives the filtrate. So let me show you another picture and show you exactly what this is. So if you look here, right, this ball of capillaries right here is called the glomerulus, guys. So that's the glomerulus. And then this capsule, this yellow capsule that goes around, that completely surrounds around, sorry, red was not a good color here, but completely surrounds the glomerulus is what we call the glomerular capsule or Bowman's capsule. You'll hear both terms used, glomerular capsule or Bowman's capsule. And I will try to make sure that I use both terms on the exam, okay? And so those two together make up the renal corpuscle. So again, these two, this whole unit here is the renal corpuscle. Let me see if I can change my pen color here just to make this a little more obvious. Let's see if I can find no, that's not going to be different enough. Let me find a color that might show up just a little bit more. Probably not blue. How about let's try magenta. Okay, so this entire structure here, okay, is the renal corpuscle. So again, renal corpuscle consists of the glomerulus, and the glomerulus is the bunch of capillaries. Guys, for some reason, this always screws students up, okay? The glomerulus is all these capillaries, okay? So it equals all those capillaries. And then the glomerular capsule is that part right around it, okay? So, and then just to give you an idea, so that's the renal corpuscle, and then everything else, all of these tubules, okay, the proximal, you can see this is called the proximal convoluted tubule, into the loop of Henle, and then the distal convoluted tubule, okay, and then here the collecting duct are all part of the renal tubules that make up the nephron. So those are the two parts of the nephron, again, the renal corpuscle and the renal tubules. Now, when we talk about the renal corpuscle, the glomerulus, those capillaries, okay, are going to receive blood from a very large, remember, afferent arteriole. So remember, the afferent arteriole is bringing blood in, and then it's moving through the glomerulus, and then passing it back into a smaller efferent arteriole. Now, this is important to remember, that you have this large, oops, sorry about that. You have this large afferent arteriole, okay? I'm going to over-exaggerate it, right? And then dumping into these capillaries, right? Okay? And then, okay, coming back out, a smaller efferent. That was not a great picture. Let me redo this picture here. So if we were really going to draw it more accurately, we would have a large afferent arteriole, okay? And then it would branch into your lovely capillary bed here, okay? And then it would finally, your capillaries would finally dump into, okay, a smaller efferent arteriole, okay? And so, and that's what this is supposed to be. So this would be your afferent arteriole, okay? And you can see it's larger. And then here would be your efferent arteriole, which is much smaller. And this is the reason that this is important is this creates kind of the effect almost like a hose. So if you look here, what ends up happening is you end up, it's kind of acting like the nozzle on a hose, and you end up building up a bunch of pressure because this afferent arteriole is bigger than the efferent arteriole that's exiting, okay? So as blood is moving through here, it really slows it down in the capillaries here, okay? And it's slowed down because you get a backup here because the, again, diameter of your efferent arteriole is smaller than your afferent arteriole. So that's very important, and what it does is it creates a high pressure in these capillaries. And that's important because that high pressure is going to help to push fluid out and into the glomerular capsule, which is going to be surrounding this. Wow, my drawings are horrible today. Sorry about that. So there would be this capsule that goes around here. See if I can color that in. And then this capsule that goes around the glomerular capillaries here. And so again, this wide afferent arteriole coming in, okay, is then going to act as a hose, and then the efferent arteriole is going to act as a nozzle. And what it's going to do is you're going to build up a bunch of pressure then in these the glomerular capillaries, and that's going to force fluid out into the Bowman's capsule. So that's how the filtrate is going to be formed in just a minute. We're going to recover this in just a minute, but I wanted to explain why it's important to understand that the afferent arteriole is larger than the efferent arteriole is in diameter. Okay? So really important, hint, hint, very important to understand that. The efferent arteriole will then eventually give rise to what are called the peritubular capillaries. So in this drawing here, you can see here's your afferent arteriole going into the glomerulus and then branching back into the efferent arteriole, and then the efferent arteriole here is going to branch into these peritubular capillaries that you see completely covering all of the renal tubules here. Okay? So that's what the peritubular capillaries are. Okay, so let's talk a little bit more now about the glomerulus itself. So the glomerulus is this ball-like tuft of capillaries, okay, in here. And again, you can see that in here. And so let me orient you in this picture. Here's your afferent arteriole coming in, and then it branches, okay, into all of these little capillaries that we call the glomerulus. And then eventually they come all back together to form the efferent arteriole, so the blood can then leave. So literally blood flows, boom, through the afferent, oops, I'm sorry, hit my button there. So it flows through the afferent arteriole into these capillaries and then out the efferent arteriole, okay? And it's at the, again, the glomerulus where filtration is going to happen, okay? Where we're going to start to filter, and that's what the glomerulus basically is a big filter. This is where we're going to start to filter, again, things into the nephron. So what's going to happen is water and solutes are going to be filtered through the glomerular capillaries, okay, as blood passes through. And so what will happen is, again, remember that we've created this high pressure situation because we've got this large afferent arteriole and small efferent arteriole. So we created this high pressure situation. So what happens is as blood moves through here, okay, now you're going to see that these capillaries are leaky here, and they're going to leak out water, okay, because water is going to push its way through because the pressure is so high. So water along with certain solutes like dissolved ions that are in it, okay, and they're going to push them out from the capillaries, okay, into the Bowman's capsule, okay. This is the Bowman's capsule or the glomerular capsule right here. Okay? And then from there, all of that fluid will then move into the proximal convoluted tubule. Okay? So let's talk a little bit more about how, again, the glomerulus acts as a filter. And we'll get into the structure of the glomerulus and how it acts as a filter in just a minute here. Real quick, I just want to talk about, while we're here, this thing called the juxtaglomerular apparatus. At the point of contact between the afferent and the efferent arterioles. And then we're also, this is going to be one of your renal tubules that we haven't talked about yet, and also the distal convoluted tubule, which will make sense when we get there, okay? The epithelial cells of the distal tubule form what's called the macula densa. So let me show you a picture of this. Here you can see this right here is called the macula densa. Okay? And then here we have these juxtaglomerular cells right here that are part of the afferent arteriole. So the juxtaglomerular apparatus is going to be this whole area here where you have the distal convoluted tubule that's in between the efferent arteriole and the afferent arteriole. Okay? And so this area here and all the tissue here is going to make up the juxtaglomerular apparatus. So here you're going to see there are cells here and then here. Okay? And this is called the macula densa right here, are going to make up the juxtaglomerular apparatus. And that's going to be very important for controlling the blood pressure within the glomerulus. So near the macula densa, okay, on the afferent arteriole are going to be the smooth cells called the juxtaglomerular cells. And those are going to be right here, guys. So here on the afferent arteriole are these juxtaglomerular cells. And here on the distal convoluted tubule are going to be the macula densa cells. Okay? And together, since they're right next to each other, they butt up to each other, they're called the juxtaglomerular apparatus. The macula densa, again, together with the juxtaglomerular cells, makes up the juxtaglomerular apparatus. And so oftentimes we abbreviate this as JGA. And the JGA, or the juxtaglomerular apparatus, is going to help monitor and help regulate blood volume and blood pressure through this, what we call renin-angiotensin mechanism. Now, we're not going to go over what we call the RAS mechanism or the renin-angiotensin mechanism because it's it gets a little bit involved. It's a cascade pathway. But what I want you to know is that this juxtaglomerular apparatus is going to monitor and regulate our blood volume and blood pressure of our blood that's entering into the glomerulus here. So that's kind of important to keep in mind. So again, this is kind of nice because this breaks down the whole, again, the renal corpuscle and some of the other attached parts here. So here you can see there's the afferent arteriole, right, coming into the glomerular capillaries and then out the efferent arteriole. Here again, surrounding the glomerular capillaries is the Bowman's capsule or the glomerular capsule. And then here you're going to see this is actually a later part of the nephron, one of the tubules. So this is going to your proximal convoluted tubule will then lead into the loop of Henle, and then as the loop of Henle comes back up, this is actually the ascending limb of the loop of Henle here. And here's where you have the cells of the macula densa, and then here you have your juxtaglomerular cells, and together these cells make up the juxtaglomerular apparatus. Okay? So again, that just gives you a little review of kind of the overview of the juxtaglomerular apparatus. So again, the structure where the afferent arteriole makes contact with this ascending loop of Henle right here, okay, is where that's our, again, juxtaglomerular apparatus. So the macula densa, we have the macula densa here, which is that thickened portion of, again, the loop of Henle. And then we also have the juxtaglomerular cells here, okay, which are just these modified muscle cells here that are in the arteriole. And again, it helps to regulate blood pressure and blood volume. So again, I just tried to give that to you a couple different ways so you can see what that juxtaglomerular apparatus is and why it's important. So the nephron, again, is that functional unit of the kidney. So what we're going to do now is we're going to talk about how the nephron makes its urine. And for the first part of the lecture, we're going to talk about number 1. And then I'm going to break this up, and in the second part of the lecture, then we'll go into number 2 and number 3. Okay? So again, I'm going to split this lecture just so that it's not so large that it's hard for you guys. I know it becomes challenging then for you guys to download, and it's getting hung up. So hopefully by breaking this into 2 lectures, it'll make it easier. So we're going to first talk about the fact that the blood is actually filtered, okay, of small molecules and ions in a process that we call glomerular filtration. That's the very first part of the step, is forming the filtrate. So that's going to happen in the renal corpuscle. This is where, again, water and certain small molecules and ions are moving from the glomerular capillaries into the Bowman's capsule here. Okay? And that, when that happens, that point at which that fluid enters the glomerular capsule, now that fluid is called filtrate. Okay? So once it's entered here, we call it filtrate. So then, as that filtrate moves through the renal tubules, okay, you're going to see that we reclaim some of the needed amounts of useful materials, certain ions, things like sodium, that are going to be needed. And so they're going to actually be reabsorbed. So we call this tubular reabsorption. What that means is we're reabsorbing the things that we need back into these peritubular capillaries. Okay? And so that'll be the second part, tubular reabsorption. And then the third part is that surplus or waste molecules and ions that are left will flow out as urine, and that's tubular secretion. So again, anything that's left in here after tubular reabsorption has happened will then be released as urine. Okay? So that will be our waste molecules. And our waste, okay, again, the expulsion of that waste will be our tubular secretion. We're getting rid of that surplus or those extra waste molecules. Okay? So again, those are the 3 main parts. glomerular filtration, okay, so the process of actually making the filtrate, then tubular reabsorption, where we take back what we might need, okay, that was filtered out up here in the renal corpuscle, and then there's tubular secretion, again, getting rid of the excess waste and the surplus of ions and things like that. Okay, so again, we're going to focus on glomerular filtration for this portion of the lecture. So for glomerular filtration, this is the very beginning where we form our first filtrate. So urine formation actually begins here when the fluid portion of the blood is filtered by those glomerular capillaries called the glomerulus, and it enters the glomerular capsule, and just like I said, now we call it glomerular filtrate. So let me show you what this capsule is going to look like and how it acts as a filter. There are extensions of the capsule that are going to be called podocytes. And these podocytes are going to be extensions from the capsule that surround the glomerular capillaries, or the glomerulus. So let me show you what this looks like. So here you can see, here in red here are the capillaries that make up the glomerulus, okay? So remember, when we use the term glomerulus, glomerulus means capillaries, okay? So again, I want you to think glomerulus means capillaries, okay? So now, if you look, the glomerular capsule, right here, this outer portion here, capsule, is going to have these little extensions coming off of the cells that will actually surround the glomerular capillaries, just like this. So you can see here, and these are what we call podocytes, okay? So here you can see all of these extensions from the capsule cells, the glomerular capsule cells, that extend out into these little foot-like structures that we call podocytes. And they completely surround the glomerular capillaries. And this is where filtration is going to happen. So, and you're going to see that you're going to have these little slits in between the cells that we call filtration slits. So this is where again, water along with certain solutes and ions are going to move from the capillaries, okay, and then through these filtration slits and then into the glomerular capsule here. So, again, we're going to talk a little bit about pressure, and I kind of already talked about this, but why it is that we are able to move water and solutes out of the capillaries and into the glomerular capsule. And the reason is that we have this pressure that's built up in the glomerulus. And the main force that's responsible for pushing fluid out into the glomerular capsule is going to be simply hydrostatic pressure, okay, of the blood inside. And so remember again that we have our afferent arterial, right, branching into those capillaries, and then exiting, okay, eventually our efferent arterial, which is much smaller. So again, what we've done is it acts literally like the nozzle on a hose. And what it does is it creates a lot of hydrostatic pressure. Just like, again, if you put a nozzle, especially a nozzle with a more narrow diameter, right, on your hose, then it's going to create the stream of high pressure water, right, that's exiting the hose. So the same thing is happening in the glomerulus. Again, we have a large afferent arterial and a small efferent arterial, and so what ends up happening is you have these little capillaries here in between, and this creates a lot of hydrostatic pressure, okay, inside. And hydrostatic pressure is just the pressure of that fluid, okay, the fluid and blood pushing out on the walls of the capillaries. Okay? So that's your hydrostatic pressure. And hydrostatic pressure is high inside these capillaries. So that's the main pressure that's going to push fluid out. Okay? Now, due to certain plasma proteins, especially albumin, remember we talked about albumin being one of the main proteins in your blood plasma. So it has a main purpose, and one of its main purposes is to help maintain osmotic pressure so we don't lose too much fluid from the blood, okay? So, our plasma proteins are going to create an osmotic pressure of the blood that's going to resist filtration, okay? As does what we call our capsular hydrostatic pressure. So, and let me kind of show you, I'm just going to show you what this is on a picture, okay? So, we have our first force, and I'm just going to break this down because I know that this gets a little bit confusing. So, I have this here verbally for you, but I want to break it down for you and try to make it a little bit easier over here. So, this is the deal with this. We basically have, okay, these three main pressures here. Okay, we have glomerular blood hydrostatic pressure. We'll just call it hydrostatic pressure or glomerular hydrostatic pressure. We need to say the glomerular hydrostatic pressure, and you'll see why in just a minute. So we have our glomerular hydrostatic pressure, okay, GHP, and that's going to be the pressure that is pushing fluid from the glomerulus or from the glomerular capillaries into the capsular space here. Okay, so that's pushing one way, right? Now we have two other pressures that are going to help keep fluid in the blood so that we don't lose too much fluid from the blood into the glomerular capsule. Okay, and so these two pressures that are pushing again in the opposite direction are going to be capsular hydrostatic pressure. So here's capsular hydrostatic pressure. And what that is that's just as fluid builds up in here, it's going to create its own hydrostatic pressure pushing back on the capillaries and preventing more fluid from leaving the capillaries. And then we also have what's called our blood osmotic pressure. Okay, so blood osmotic pressure. And what that means is that there are certain solutes like albumin, okay, inside the blood here, and they are, remember that when we have an area of high solute, water wants to move down its concentration gradient into the high solutes. So that blood osmotic pressure is going to help draw back in some of that fluid, okay, back into the blood. So what that means is, okay, we have one pressure, glomerular blood hydrostatic pressure, pushing fluid out into the capsule, and then these two other pressures kind of drawing fluid back in, capsular hydrostatic pressure and blood osmotic pressure. Okay? Now, okay, the deal is that glomerular blood hydrostatic pressure is the big guy. If you could picture again, these three football players, okay? The one football player is pushing in one direction and the other two are pushing in the other directions, okay? So here you've got glomerular hydrostatic pressure pushing this direction, okay? And then you have the capsular hydrostatic pressure and blood osmotic pressure pushing this way, okay? But these two guys are smaller than this guy. Okay, meaning glomerular blood hydrostatic pressure is greater than these two combined, okay, that are pushing in the opposite direction. So you're going to get a net movement, okay, in this direction. We just won't lose too much. So let me put some numbers to this, and this might make it a little bit easier for you, okay? So the glomerular hydrostatic pressure here is going to be about 55, okay, 55 millimeters mercury. So 55 is going to be your pressure here, okay? Now, capsular hydrostatic pressure and blood osmotic pressure that are pulling fluid back into the capillaries, okay, are going to total only about 45 here. So again, capsular hydrostatic pressure is 15 and blood osmotic pressure is 30, okay? So together, they are only equal to about 45, okay, millimeters mercury pressure. So, okay, again, we're going to get a net filtration or a net pressure of 10 millimeters mercury pushing fluid out into the capsule, okay? So, what that means is we have a net loss of fluid. It just means that we're not losing too much fluid, okay, if that makes sense. So again, we have glomerular hydrostatic pressure pushing one direction, capsular hydrostatic pressure, blood osmotic pressure pushing the other direction, but glomerular hydrostatic pressure is greater than these two pressures. And so we get a little bit of filtrate again moving through here. And it's literally our perfect amount of filtrate that we can form. So those are the processes that are involved. Those are the processes that are involved with forming the filtrate. And again, it's important to understand this because without capsular hydrostatic pressure and blood osmotic pressure, we would be losing way too much fluid into the nephrons. Okay, so these two pressures help prevent us from losing too much fluid from the blood. Okay, so let me see if I can get rid of my drawing there. So again, this is just a summary. You have your net filtration pressure, okay? Your net filtration pressure is going to be the total pressure that helps promote filtration. So that's going to be this pressure here. And the net filtration pressure is going to equal your glomerular blood hydrostatic pressure, okay, minus your capsular hydrostatic pressure and your blood osmotic pressure, okay? So again, hopefully that all makes sense. The nice part about having you in class is I can usually look at your faces and see if you're getting this. If you're not getting this concept, please, please, please contact me and I can maybe dig up some animations for you, something so that you understand this process because this is very important to the formation of the filtrate. So filtration rates, okay? The factors that affect the filtration rate or the rate at which we're going to be making filtrate in the glomerular capsule, okay, are going to be your net filtration pressure. Okay, obviously whatever your net filtration pressure is going to impact the rate of filtration. The glomerular blood osmotic pressure, so that's your BOP, okay? Your BOP, or blood osmotic pressure, plasma osmotic pressure, blood osmotic pressure, same thing. Sorry that I swapped words on you, but again, it's the same thing, blood osmotic pressure, okay? And the hydrostatic pressure that's in the glomerular capsule. So basically, what affects your filtration rates? Okay, all of these pressures. Okay, all of these pressures affect your filtration rate. When the afferent arteriole, okay, because remember that arterioles are going to have a layer of muscle. Remember we went through the different tunics? So remember you had your tunica externa, tunica media, and your tunica interna, and the tunica media consisted of muscle cells, smooth muscle cells. So around the afferent arteriole are these smooth muscle cells, and they can constrict. And if they constrict in response to sympathetic stimulation, then the filtration pressure and thus the filtration rate will decline. And so let me go back up and show you why that is. Okay, so if your afferent arteriole constricts here and becomes smaller, let's say it constricts to, maybe about this size, now not as much blood is moving through here into the glomerular capillaries. And that means that your hydrostatic pressure, less blood moving through there, less fluid, less hydrostatic pressure. Okay, so that will influence our hydrostatic pressure. If our hydrostatic pressure, our blood hydrostatic pressure, I should emphasize, so that's our glomerular hydrostatic pressure decreases, then again that's this. If this decreases, then our overall filtration pressure decreases. And that means that less filtrate will be formed, okay? Because we're not pushing out as much fluid into here. So again, that's important to remember as well. When the afferent arteriole constricts, our overall filtration rate, as well as the actual amount of filtrate that's formed in that glomerular capsule, decreases. When the efferent arteriole, so when this arteriole here constricts, then it's just the opposite, guys. Here now, okay, we've made the nozzle on the hose even smaller, and that builds up extra pressure here, okay? More pressure than normal. And so that filtration pressure is going to increase, and that's going to increase the rate at which we're going to be pushing, again, fluid out into the glomerular capsule. Okay? So again, let me just kind of review this in the picture. So if our afferent arteriole constricts, okay, then glomerular blood hydrostatic pressure goes down, and our overall net filtration goes down, okay, and so we have less filtrate being formed. So filtration declines. Then, okay, if our efferent arteriole constricts, but our afferent arteriole stays the same, okay, our efferent arteriole constricts, then that's going to create even more glomerular blood hydrostatic pressure, okay? So that's going to make this even greater. And if this number is even greater, okay, then our net filtration pressure will be higher and more filtrate will be formed. Okay? So again, you know, and that may be something you just want to make a note card of. Hopefully it can become intuitive for you, but that's something that you definitely need to know. Again, if the afferent arteriole constricts, net filtration decreases. If the efferent arteriole constricts, then your net filtration increases. Okay, so hopefully that helped clear that up a little bit. When osmotic pressure, okay, in your blood, so in the glomerulus, is high, the filtration rate is going to decrease as well. So, okay, let's say that you know, you are dehydrated. Okay, if you're dehydrated, then you have less fluid per solutes, okay, inside here in your blood, and what that means is you have more solutes. Okay, and if you have more solutes, that means that your osmotic pressure is going to be higher. Okay, so it's going to want to pull more fluid back in. So, okay, if this pressure is higher, okay, so again, this is your blood osmotic pressure right here. If your blood osmotic pressure is higher, okay, then here remember that this is subtracted from your big hydrostatic pressure. So if this is higher than it normally is, then this number is going to be lower, okay, than it normally is. So again, blood osmotic pressure is going to cause you to want to pull more fluid, suck more fluid back into those capillaries, and that's going to decrease the overall filtration rate and the amount of filtrate that's formed in here. So again, these are all things that affect the filtration rate. Oops, sorry about that. When hydrostatic pressure inside the glomerular capsule is high, filtration rate is going to decline as well. So again, we're just playing with all these pressures. So if, let me go backwards again just to show you this picture. If your capsular hydrostatic pressure is high, okay, if this is high, then you're going to be, again, pushing more fluid back in here, and that is going to decrease your overall net filtration. Okay? So if this increases, then your net filtration is going to decrease. Okay? And your filtration rate declines. So again, all we're doing is playing. Guys, if you know this right here, okay, then you can solve any question, right? Because that way then you know if something makes the hydrostatic pressure increase, then net filtration increases, okay? If, on the other hand, something instead increases the capsular hydrostatic pressure, okay, since that's subtracted out, that's going to decrease your overall filtration rate. Or if something increases your blood osmotic pressure, that's going to decrease your overall filtration rate, okay? So again, it's just, that's basically all this is doing is talking about all the things that impact the filtration rate and impact that equation that we just talked about. So on the average, oops, too far. On the average, the filtration rate is about 125 milliliters per minute, or 180 liters, okay, in 24 hours. You may say, wow, okay, 180 liters is quite a bit. Why am I not urinating more? Okay, and the reason is that most of this is reabsorbed further down the nephron. So remember, this is all happening just in the renal corpuscle, okay? Then when we get into the renal tubules, most of this is actually reabsorbed, okay? So don't worry, you're not going to be losing 180 liters in 24 hours, right? Okay, so the glomerular filtration rate is usually relatively constant, although sympathetic impulses may decrease the rate of filtration. And the reason, remember, that sympathetic impulses are going to decrease the rate of filtration is that sympathetic impulses are going to contract the afferent arteriole. Okay, and when that happens, again, we decrease our glomerular blood hydrostatic pressure and decrease our overall filtration. Okay, and so this is kind of cool, guys, because this ties back into when we talked about, remember, the sympathetic nervous system results in a decrease in urine output. So again, one of the reasons is that we are actually decreasing our filtration rates with the sympathetic nervous system. Another control over the filtration rates, again, in the renal corpuscle, is something called the renin-angiotensin system. And this actually is going to be more involved with regulating our sodium excretion. So when sodium chloride, or again, sodium chloride being one of our salts, one of our main salts, when the sodium chloride concentration in the tubular fluid decreases, okay, the macula densa will actually sense these changes, and it will cause the juxtaglomerular cells to secrete a substance called renin. Okay? And the secretion of that renin is going to trigger a number of reactions that we're not going to get into, okay, because that's Bio 202, that leads to the production of what we call angiotensin 2. And that angiotensin 2 is going to act as a vasoconstrictor, okay? And that, in turn, is what's going to affect your filtration rate, is, again, that angiotensin. So the presence of angiotensin 2, okay, is also going to increase the secretion of another hormone called aldosterone, okay? And that's going to stimulate the reabsorption of sodium. So again, this, I'm not going to ask you, honestly, a lot of questions about the renin and angiotensin system, just because we don't have really time to get into it, unfortunately. But I do want you to know that this regulates our sodium excretion. Okay, it's very important for regulating the sodium excretion. And the heart can also increase our filtration rate when any time that blood volume is high is another way. So again, like I said, you don't have to know the kind of the nitty-gritties of this because it really gets a little bit more into depth than we have time to get into. But I just want you to know that this renin-angiotensin system is another way in which we can control filtration rate through regulating sodium excretion, okay, without getting too much into it. And then, lastly, also the heart can also increase the filtration rate when the blood volume is high. Okay, so again, the heart also has some control over that in terms of, again, it's controlling, you know, how much blood volume we're pumping out. So, and this shows you the complexity of this renin-angiotensin system. So again, this renin-angiotensin system is very complex, and like I said, unfortunately, we just don't have time to get into this. But if you take Bio 202, you will get into this, and we go into it in quite a bit of detail. But I just wanted to show you how complex really that renin-angiotensin system is for controlling filtration rates. Okay, so what I'm going to do is I'm going to go ahead and I'm going to stop here. This is a good stopping point, and then we'll pick up tubular reabsorption here in just a little bit. So, and you can, good timing, you can hear my dogs barking there. So again, this is where we'll stop the lecture, and then we'll pick this back up in the second, part 2. So, and you can watch part 1 and part 2 together, but you might want a little break, which I don't blame you.